So I'm going to talk about, this is an ambitious title, Quantitative Genetics of Protein Glycosylation, but I will try to measure up to the title. Right, here is my disclaimer. So the thing I should say is that since recently I'm an employee of GSK, but this presentation is my personal opinion and not that of GSK. Well, basically this work was done before I joined the company. Right, so my talk, I'm going to have three parts to it, and I start with, a little bit of propaganda of genetics and genomics, right? I'm a geneticist, and I always, take my chances if, I'm allowed to promote this wonderful field of study. And then I'm going to review 12 years of genome-wide association studies of human protein and glycosylation. I think that started with, with Gordon's papers in 2010, so we are roughly 12 years into this area. And I was lucky that I was invited early on board. I think we already worked in 2012 on the first IgG and glycosylation GWAS together. Right, and the third part is conclusions and discussions, obviously. Good. So if you think of this, right, humans are terrible study objects, right? And this is why we have all these wonderful models. We have mice, and now we have cell lines, and organoids. But still the models, they're called models because they are not capturing the full complexity of what is happening in humans, right? And of course we all would, in an ideal world, you would like to have your biology done directly in the object you're interested in, right? And many of us are directly interested in human. But the problem is that the experimentation, right? You cannot really do good experimentation with humans. Now, the idea is that the idea which, is unique to genetics and genomics is that we can follow the experiments of nature, right? We have this naturally occurring genetic variation, and if you look into our genomes, you will see genes which are knocked out, the genes which are knocked down, the genes which are conditionally knocked down, right? So I mean the total loss of function mutations or mutations which up or down regulate the expression of specific genes or the expression of the proteins, right? And actually variants which would up or down regulate the expression in specific tissues, right? So you find all of this. Of course, there is a little bit of catch, the things which are too important to be knocked out, you don't quite see unless in heterozygotes, right? Just because this never makes it to the stage when you can study it. But, these complications aside, genetics provides this unique and wonderful way to study human biology in vivo, in humans, right? And this is totally unique, I think. Right. Now, I'm going to talk about a forward genetics approach, and forward genetics is, again, a wonderful thing, because to do forward genetics, to study biology with forward genetics, you basically need only two things. First of all, you need to be able to measure genome, right? And we can measure genomes very, very well, right? We can sequence them, or we can do array, etc., etc., etc. Now, the only one other thing which you need is you should be able to measure your phenotype. And that's it. As soon as you can measure genome, and you can, and as soon as you can measure the phenotype, right, you can apply forward genetics. So what you would do, you would go, along the genome and see this naturally occurring, variants, inherited variants, standing genetic variation, right? And we will try to relate, to associate this populational genetic variation to the variation of your trait, right? And in this way, you are going to detect, regions of the genome where variation is affecting the trait of interest, right? So this is almost, technical way to study anything as soon as you can measure this anything. So far, so good. I'm going to talk specifically about population genetics of N-glycosylation. So we've been studying large cohorts of healthy people, right? As soon as, people after 40 could be healthy. We've been studying healthy people. And therefore, I will spend, a couple of minutes telling a few words about how the GWASs are done, because, well, this is quite technical, but I think it brings some important message and limitations of the approach. Definitely there are limitations of the approach. So what you do, okay, I mentioned you measure the genome, and now I'm concentrating on, say, specific polymorphism, specific site in the genome where two alleles are segregated in the population, and I, call them A and B. So I have three genotypes in the population. Some people, AA, some people heterozygous, some are BB. Right. So what I do, I go to the population and I find a person. What I consent and everything, and ethical approval, etc. But what I do, I measure the phenotype of interest, right? And I measure this phenotype, and I measure the genotype, and then this first sample I put here, right? So this is, say, trait value of this person, and it's homozygous, right? Okay, this is not GWAS yet. I need more things, right? So I go and, find more participants to my study. So next one is heterozygous, slightly lower value. The third person, again, homozygous. Well, I need many points. When I do GWAS, I use very large samples, right? But after a while, when I connected, thousands of samples, I can really see if, the genotype change the value of the trait, right? And most of the time it doesn't, because most of the genome does not contribute to the variation of your trait. But sometimes you see this trend that substitution of certain allele, B or A, does affect the mean value of the trait. So you say, I'm finding association here. And you do this for, millions of markers spread throughout the genome. For each marker, what you do, you compute the regression coefficient, you compute p-value. I'm making it a little bit too simplistic, perhaps, because there are way more complicated methods, multivariate analysis, mixed models, etc., etc. But, essence is very simple. You see if there is association between genotype and the phenotype, right? And then you put this dot on the graph, and here, is your negative log p-value, right? The higher, the stronger is statistical association. And here is the genomic coordinate of this polymorphism, right? And you do it for your millions of SNPs, right? And when your statistical significance is high enough, you say, oh, I detected association. So now I will go down this, peak and look what is below there, which perhaps will give me a clue how biologically this locus is working. So one important thing to realize is that when you do this part, you are detecting loci, and loci may contain many, many genes, right? So you actually do not know which specific gene's function is, disturbed and affected. It could be, gene here under this peak, but actually it could be regulatory element enhancer, which acts, 2, 3 MB apart. So this is just beginning of your story, and you really need to, work out your hypothesis, how this variation, how functionally it works, and what is the gene affected. Although in this case, perhaps when I talk about glycan seen GAT3 is kind of making story perhaps straightforward. But it's not the case most of the time. So what you normally do, you have so many loci, so many hypotheses. If you will try to do like classical experimentation on each of these genes, you will never finish, right? So you try to narrow down your hypothesis by combining functional genomic data and a lot of, computational systems biology and computational genomics techniques to work out good hypotheses. What are the best candidates in the region? And when you have candidates and you think it's worth its while to, follow it up in laboratory, then definitely you want also to have your experimental proof for this specific gene. Okay, so this is like basically outline how things work through genetics and genomics, how you uncover biology and regulation through these approaches. I'm not going to talk about importance of glycosylation, like no point here, and it's great. One of the great things which I thought, Gordon kind of convinced me to join this story is that we know a lot about like biochemistry of N-glycosylation, right? And this gives an edge because you kind of understand how things work, who are the workers in this story. But then the thing is that, of course, this is regulated, right? And there is tissue-specific regulation, so glycome of one tissue is unlike the glycome of other tissue, and there is also inter-individual variation. And so it's very much, although we all share, and all our tissues share kind of genes encoding the machinery, the regulation of this machinery, tissue-specific, but also between people, makes the story quite unique, right? So what we are interested in is, this big regulatory story on the top of the workers. Okay, so our questions are: which tissue-specific networks of genes determine inter-individual differences in human protein glycosylation? And quite importantly, because we see many associations between complex disease and glycosylation, but we don't quite know how it works. Often we don't. Therefore, the second question we wanted to address is, what's the nature, what is the mechanism behind this association? And here genetics and genomics, again, is quite good too, because this backbone of variation allows you to put together information from really different studies, different domains. Okay, so what have we learned in 12 years of genome-wide association studies of N-glycosylation? Well, first of all, the traits we measure, we can put it as simple as, the heights of the peaks on the chromatogram, right? So we are studying a large number of traits which reflect abundance of different glycan structures. And it could be, N-glycans attached to immunoglobulin G in one case. And actually, this is the most studied story, right? Because there have been exactly 5 studies published on GWAS of immunoglobulin G and glycosylation. And the sample sizes were rather big, up to 8,000 people. And we have detected 29 and replicated 29 loci associated with variation of IgG and glycosylation. But that could be also, total blood plasma glycome, which sounds like a messy thing to do, but still, it's very interesting, and I will actually talk about it later. So you chop off all N-glycans from all blood plasma proteins. You look into chromatogram, you have again a bunch of traits, you study it. These studies have been smaller, it's only up to 3,500 people, and about 15 loci were detected. And then very recent development this year, GWAS was published of transferrin and glycosylation, only 2,000 people, you can say only 8 loci, but I think it was a huge advance which allowed some, conceptual changes. So altogether, if you add up, this is like 50, but in fact it's 34 loci because you see the same loci popping up on different glycomes. Okay, so here are these 34 loci, and I named them by the candidate gene which we prioritized in this locus, right? So it's not, it's not carved in stone which one is the candidate gene, but that's our best guess about possible candidate. And when you do it for many loci, you kind of start seeing some pathways, which I'm going to talk about a little bit later. Right, so these are the 29 loci which we detected on IgG and glycosylation studies. They overlap quite a bit with 15 loci which were detected on total plasma and glycosylation studies. And finally, this red one is the loci which are detected in transferrin. So why I'm saying I think this transferrin story was a very important conceptual advance is because, for example, you see that, look, there is no plasma loci, right? You can always classify a locus either to IgG, so this part, or to transferrin, right? And this kind of makes sense because if you think of glycoprotein composition of blood plasma, the two major contributors are liver and bone marrow plasma cells, right? So you could think, at least in the first approximation, it seems we can see total plasma and glycome as a superposition of glycomes driven by glycosylation processes in liver and in plasma cells, right? And moreover, by availability of both IgG and glycosylation jivas, and now transferring glycosylation jivas, any locus which we can detect now or in the future plasma and glycosylation studies, we can look, does it have the same pattern association, this locus, as with transferrin or IgG? So we can classify this loci very neatly, right, into, well, like I would call it liver and plasma loci, right? It may be too much of generalization, but otherwise I'm repeating all the time, IgG-like association pattern or TF-like N-glycosylation pattern. Okay, so again, I think this was very important advance in terms of understanding and resource, right? Okay, the next thing which was quite remarkable is that the overlap between, the loci which are acting both in, well, I'm going to say liver and plasma cells, but please remember I'm always saying which pop up on IgG and glycosylation jivas and TF glycosylation jivas, right? This overlap is rather limited, right? And I think if you look at this, perhaps for some of you, it maybe even doesn't make sense. For example, like, okay, ST6-GAL1, B4-GALT1, right? How could that be that it's IgG locus, right? So everybody knows it works in liver. So this is this conditional knockout thing, or knockdown thing, right? So while both of these glycosyl transferases are expressed both in liver and in plasma cells, the genetic variation is apparently in the element which is regulating its expression only in plasma cells, right? So that's an example of conditional knockdown I was talking about. Okay, I'm not going to dig too much into expression because I think I'm getting over time very soon. Right, so if you try to look into, like, what are these genes? What are these genes which we detect, right? Well, first of all, here in blue are 9 glycosyl transferases. I think what is maybe interesting, or at least it was interesting for me, these are glycosyl transferases which are acting in medial and trans Golgi. We don't see this earlier, right? Then second large group are transcription factors. Transcription factors, and most of them are tissue specific. So we have a bunch of transcription factors which are, like RUNX1, RUNX3 somewhere here, Icarus first, Icarus third. So these are like B cells transcription factors, right? But then we look into this liver part, and you see HNF1 alpha, which is like one of the liver transcription factors, right? So we see a bunch of the transcription factors, and then we see a number of other, which I will get into a little bit details in the next slide, right? But you should realize there are like 7 out of 34, like quarter of regions where we don't understand what's going on. What is the candidate? And maybe that's, the gold mine of unknown biology, right? Because what we see here is, between new biology and systematization, right? And, figuring out the relative role. But this perhaps is like really totally new biology, but also very tough one. Okay. Right, so what kind of elements we see? So if I try to compose like my view of protein N-glycosylation from the genes I see, right, where variation contributes to populational variation of N-glycosylation, these are the elements I see. So here I have like glycosyl transferase, which is acting upon glycoprotein, which is being, sometimes transported outside directly, but sometimes it's also degraded because things didn't, go well. And I have some transcription factors, which are, some of them I can see they are directly regulating, they are sitting on, like sites near to glycosyltransferases. But then I see these factors which are very important for differentiation and functioning and development of the tissue which secretes this glycoprotein. And I have some non-specific factors which are affecting like performance of glycosyltransferases in Golgi. So I have examples of any line which I draw here, like, and I have also nice examples where we have like this knockouts. For example, FAT6. There is this common allele which really kills the function of the protein, right? And we have examples of knockdowns where it's expression going up and down. We have examples of conditional knockdowns, as I mentioned. And transcription factors, things about the tissue differentiation, but also variation in the glycoproteins in question, like transferrin. We see that again, there is a relatively common structural mutation of transferrin, and then it's associated with change spectrum of its glycosylation. All right? And non-specific factors like this SLC9A9, which we believe works through change in Golgi pH. Okay, and then things about transportation and degradation before you can measure it in your plasma. So that's kind of view which appears, and I realize I'm missing, many parts of the picture, but right now with genetics, we didn't see it, right? I think when we go deeper, we will. Okay, overlap with the disease. I should round up, therefore I only mention an interesting observation which, I mean, we should be able to substantiate in the next round of GWASs. But it seems that when we see overlap between glycosylation-related loci and autoimmune and inflammatory disease, this is like big category, this is the loci which are expressed in plasma cells, which makes sense, right? And interestingly enough, cardiovascular disease and its risk factors, so that's very much about LDL actually, right? We see overlap with the loci which are expressed on liver and glycome. Well, I should say again, transferrin and glycome, right? I may be generalizing too much again. Okay, conclusions. I hope I convinced you that genetics and genomics is a unique approach to, study human glycobiology in vivo. It has its limitations, but it's quite unique in its capabilities. Variation in quite a few loci is associated with changes with human M-glycome, right? We see that plasma M-glycome is driven by glycosylation in lymphoid and hepatic tissues. Okay. What we see is that some actors, glycosyltransferases, many regulators, and majority of genetic determinants of variation are tissue-specific. And we think that now our approach will start to bring new clues into understanding of overlap between glycosylation and specifically cancer, cardiovascular, autoimmune, and inflammatory disease. These are categories which we see enriched. Okay, in future, we strive to study, more individual glycoproteins. It's definitely going to bring more and more value, right? But also bigger GWASs are going to bring more fruits. So this is the latest published GWAS, but we are now finishing the next round, which kind of more than doubled the sample size, and you can see the difference. So the yield is more than double again. So I'm looking forward to tell you about these results, at future conference. But I need to first present it to our co-authors. Multiomics will be very important to figure out the mechanism of action of specific loci. And, well, I cannot, underestimate or overestimate, sorry, the need of, when you worked out hypothesis, you need to go and prove it, right? Okay, very good. That's about it. I want to thank all the collaborators. They are really many. It's a huge effort. Above all, Gordon, who was, who involved me into this story, but was always the driver and, someone who would always help with advice and deep understanding of glycomics. I had a privilege and pleasure to work with a group of talented, bright, motivated people who contributed to this work, my group. Well, thank you very much for your attention.